continue talking about membrane traffic. This is the second to last topic of membrane traffic. And then we'll finish up talking about membrane traffic on Tuesday, and everything up through Tuesday will be covered on the next exam. And you can look at what's covered on the exams on the syllabus. So we're going to first review snares a little bit, where we ended the last class. Then we're going to talk about the identification of signals required for sorting. Because once you leave the endoplasmic reticulum, you can go to different compartments. And we'll look at what signals on proteins determine how they're sorted. And then I hope we get to post-translational modification. Well, we definitely will, because we'll get to that. Okay, so to review what we finished up talking about in the last class, we talked about snare proteins that are on vesicles. This was just an example of one that's on a neurotransmitter containing vesicle. And to remind you that we talked about how B snares couple with T snares. Right? And we also talked about a little vignette about how Botox is based on snare biology. It is a protease that's really specific for one of these T snares. So little fun fact that you now know. So let's launch right into a Instapoll question that we talked about last time, functions for B snares and T snare proteins. And let me say we have some visitors in the back of the room. Today is Eyes on Teaching. You may experience this in other classes. So professors are invited to go to each other's classes. So they may circulate around as we do our discussions. And so be nice to them. Okay, so let's look at that Instapoll question. 5, 4, 3, 2, 1. And I have enough information from what was loaded to see that most of you, in fact, I think just about everybody picked B and C, but not A. So there's no point in discussing this, since you all could discuss the same answer. So would someone who picked E, the correct answer, explain why they picked E? So the snares and B snares are responsible not only for fusion of a vesicle with the target membrane, but also ensuring that the right vesicle fuses with the correct target. Recognize the answer, but no one wants to defend it. Go ahead.
B snare for vesicle snare and T snare for target snare. So it makes sense that the interaction between those two is the focus, and then they're specific to each other. So a B snare or T snare.
That's absolutely correct. So there are a whole bunch of B snares. What you recognized is there are a whole bunch of B snares in the cell and a whole bunch of different T snares in the cell. And it's that coupling. We talked about their cytosolic domains winding and winding together. And that gives the specificity to the vesicle, ensuring that it goes with the right target. And then what you may also remember is that winding brings the vesicle closer and closer together until those bilayers merge. So the pairing is not only responsible for the right specificity, it is also responsible for the fusion. Okay, so let's go on now. So we have begun, we talked a lot about the rough endoplasmic reticulum and how proteins begin life secretory proteins begin life in the endoplasmic reticulum. They're co-translationally translated into the rough ER. So what do you remember from previous classes, just shout it out, of what happens in the rough endoplasmic reticulum? What can happen to proteins there?
Glycosylated.
Louder?
Glycosylated.
Glycosylation. So you remember glycosylation. Perfect. And we've also covered some modifications that happen in this class. So we have glycosylation. What else? Yeah. Folding, right? And we talked about the chaperone that is one of the chaperones that's responsible for that. What was the name of that chaperone? Bip. What else?
transportation of like extracellular proteins or molecules?
So transportation of extracellular molecules, meaning that there's some budding of vesicles from the ER that are going to bring proteins that are destined for the outside to the inside. So vesicles bud from that. Any kind of proteolysis going on? We've covered one kind of clipping in depth. Does signal peptidase sound familiar? So what's that do? It cleaves off the ERSS. So these are just some of the things that, that you might remember, and you don't have to write this down because it's actually the last slide in your notes, but some of these you remember in depth. So there's, you mentioned folding of proteins by ER chaperones. BIP is one of them. There's another one called calnexin. That's just to say that there are a bunch of them. Both BIP and calnexin are soluble proteins in the ER, meaning, what do we mean by soluble is that they are not attached to a membrane but are floating in the aqueous environment of the ER lumen. There's also formation of disulfide bonds. So all proteins that are going to be in the outside world have extensive disulfide bonds. And that's accomplished by an enzyme called protein disulfide isomerase, or PDI, which is also a soluble ER protein. So why have so very few, if any, cytosolic proteins have disulfide bonds, but most of the, or all of the proteins that are going to go in the ER, I can say most of them, have extensive disulfide bonds. They also have extensive glycosylation, which you remembered, and it's a special kind of glycosylation called N-linked glycosylation that we'll have a little bit of time to talk about later. These is a preformed carbohydrate chain that's added to asparagines. So it's N-linked, and N happens to be the single letter code for asparagines. And then we're not going to cover this, but lipid bilayers are that's where all the lipid bilayers that are going to be there are covered. So the N-linked glycosylation and the disulfide bond formation are really important for extracellular proteins. And they're only found on extracellular proteins. So why? What's different about the extracellular environment versus the cytosol? You think about that. What's different about that? They're both amino acid chains, and but only the extracellular ones are heavily sugar-coated, heavily glycosylated, and tied together with disulfide bonds. And yeah, go ahead.
I was saying it's like protection against like pH.
Perfect. So protection against pH, and so the cell can control the internal environment of the cytosol, but it has no control of the outside. And it extends, and you're recognizing that pH heavily influences how a protein is folded. So the extracellular environment might experience swings in pH. It also, the cell has no control over the extracellular environment. There can be extracellular proteases out there, all sorts of things. So what the heavy sugars do, and the disulfide bonds keep that locked into a certain formation. And the sugars also protect the protein from proteases and all sorts of things. It's sort of like an M&M, where the outside hard sugar coat is protecting the chocolate on the inside, the chocolate being the protein. And this is something that's studied extensively. So not only the extracellular proteins are covered with sugar and heavy disulfide bonds, but also the extracellular domains of proteins that are going to be in the outside world. And all that is accomplished by these enzymes in the ER lumen that are, so BIP and calnexin are doing the folding, and some other chaperones. PDI is doing the disulfide bond formation, and then different enzymes are doing this N-linked glycosylation. Okay, so that's what happens in the ER, and then we have to leave the endoplasmic reticulum, and we talked a little bit about COP2 proteins, and those are responsible for budding from the ER. That's where those secretory proteins are going to go. This is a little bit of a not great diagram because it doesn't show that the COP2 COP2 code is going to fall off before this fuses with the cis-Golgi network. And then we have other traffic going the other direction, going from the cis-Golgi network back to the ER. And this direction forward that COP2 codes take is called anterograde transport or forward transport, ER cis-Golgi up to the plasma membrane. And everything going in the other direction is called retrograde transport. So we can say that COP2 codes, the vesicles, are responsible for anterograde transport, and COP1 codes are responsive, coded vesicles are responsible for retrograde transport. Now, why would you need, this seems backwards, why would you need COP1 code, why would you need retrograde transport? Any ideas? This one makes sense. Proteins are going to leave the ER and go up to the next compartment. But why do you need to go the other way? Say that again? For recycling purposes, and what do you think might be recycled? You're right. Say that again?
The snares.
The snares need to go back, that's right. So we look at how the V snare on this vesicle will fuse with the T snare. Somehow the V snares have to get back. You're exactly right. What else needs to go back? What about all that lipid? So when this vesicle, when this bilayer fuses with the cis-Golgi, eventually, if there's got to be you'd have to continuously manufacture that lipid bilayer unless you had a way to go back. And it's also the case that remember this is scooping up soluble proteins from the ER right here, and I told you that both the enzyme responsible for for formation of bisulfite bonds, as well as the chaperones, are soluble proteins. So they're floating around in here, and that liquid is captured by COP2-coated vesicles. And so with some frequency, those just end up being scooped up in COP2-coated vesicles, and the retrograde transport to the vesicles, COP1, will bring back the PDI and BIP and the other soluble proteins that are scooped up and delivered there. So all sorts of things are leaving, and then they have to come back. You might not think that these vesicles seem small, it's not such a big problem, but it actually is when you look at something at all the ER to Golgi traffic that's going on. So if we, too dark. If we look at here, this is a cell right here that's been labeled with a secretory protein right here. The slowest step of the secretory pathway is in the Golgi. So the Golgi lights up extra bright and in time, and also the ER is sort of spread, a reticular network spread throughout the cell, so it's more lacy. And if you look at these vesicles going up to the ER, there's a massive amount of vesicles at any one time. So if there wasn't a way to bring those vesicles back with COP1-coated vesicles, as you said, recycling, you'd end up losing all of the ER. You'd end up losing all those BIP and PDI proteins. It would be very inefficient. So we have a massive amount. And I can remember the first time I saw this movie, this was with GFP, and what surprised me, can you see, like, well, here you can see here. You know, we think of the vesicles as being little round things, but when you look at what's really happening, those vesicles are stretched out along tracks. So it looks like once the coat is left, they're stretched out along tracks. And those tracks turn out to be microtubules, and we'll be saying more about microtubules later in the course, how they form these tracks and their dynamic properties. Okay, so we have a problem. Soluble resident ER proteins can escape from the ER. And what are two soluble, by resident protein we mean a protein that lives in the ER that is responsible for modifications. What are two soluble ER resident proteins? Go ahead.
One of them we said was BIP.
One of them's BIP.